Welcome, fellow pastors, lay leaders, and truth seekers, to this masterclass explainer. Our goal today is absolutely clear. We're going to equip you to dismantle the secular myth of naturalistic abiogenesis, and we're going to do it using rigorous, awe-inspiring science.
That's exactly right. As church leaders, we know so many folks in our congregations feel intimidated by the secular biological narrative. So today we're diving into the hard data. We're going to turn intimidating biochemistry into a catalyst for profound worship, equipping you to show your people that our cosmos definitively points to a cosmic programmer.
Here is our game plan for today. We'll start on the street, looking at evolution's myth. Then we'll head into the academy to debunk chemical origins and unpack DNA and complexity. Finally, we'll step into the pulpit, giving you the tools to equip your church. All right, part one, the street, evolution's explanatory myth. Let's dive in. So, picture this. You're counseling a doubting college student, or maybe talking with a skeptical friend. You are almost certainly going to hear a script that sounds a lot like this. Listen, natural selection explains how complexity arose step by step. Over millions of years, tiny incremental mutations occurred. It's just science, it's not magic.
Right, we hear that all the time. But by knowing this script ahead of time, you've got a conversational blueprint. We can actually pre-bunk this whole argument just by understanding the massive category error that's taking place here.
And here is that massive, fundamental error. We have got to draw a hard line between biological evolution, which strictly requires an already existing reproducing organism, and abiogenesis, which is the origin of the very first life from non-living chemicals.
Exactly. Secular biology often conflates these two ideas. They blur them together to hide the insurmountable hurdles of abiogenesis. They try to use the mechanism of evolution to explain the origin of life itself. And guys, that is a massive chronological fallacy.
It really is. The crucial point you have to drive home is this. Natural selection is a powerful filtering process, sure, but it literally cannot operate until a self-replicating organism already exists. Non-living chemicals simply do not reproduce.
They don't pass down hereditary traits. They aren't struggling for survival. Because of that, natural selection is utterly powerless to explain how the very first cell showed up. Prior to that first living cell, you are left entirely with raw, unguided chemical chance.
Which brings us to part two, debunking chemical origins in the academy. Let's unpack the deep-level historical and scientific evidence regarding this unguided chance. It really starts with a complete collapse of early chemical origin models.
You know, history shows us this continuous retreat of naturalistic origin theories. For centuries, people actually believed life arose spontaneously, like they thought mice came from dirty rags or maggots just appeared on meat. Francesco Redi debunked that back in 1668, and Louis Pasteur definitively proved in 1859 that life only comes from life.
Yet, fast forward to 1953. The famous Miller-Urey experiment tried to resurrect the whole idea by simulating early Earth to spark life's building blocks. You'll still find it in textbooks today, but modern geochemistry has shown us it was deeply, fundamentally flawed. Here is exactly why modern science debunked Miller-Urey. First off, they used the wrong atmosphere. Early Earth wasn't highly reducing, it was neutral. Second, they used an artificial trap mechanism to protect the amino acids they created. In reality, the exact same energy, like lightning or UV rays, that formed those compounds would have instantly destroyed them. But the real final nail in the coffin is the racemic mixture problem. Listen to this. Non-guided chemistry always produces a 50-50 mix of left-handed and right-handed amino acids, but biological life demands 100% homochirality. Every single amino acid in a functional protein must be left-handed. A single right-handed amino acid completely ruins the chain, and there is no unguided natural mechanism that can sort them out. This forces a massive pivot from chemistry to molecular biology. Even if you can miraculously summon all the right left-handed amino acids, you're immediately facing an infinitely larger wall.
That's the core issue. The fundamental barrier to a naturalistic origin of life isn't actually the creation of chemicals. It's the origin of information. Life requires an incredible, totally unprecedented leap into programmatic instruction.
Philosopher of science Stephen C. Meyer developed a brilliant framework for this called specified complexity. Think about it like this. Nature can easily produce simple order, right? Like a crystal lattice using basic physical laws. And chance can produce random complexity. Think of TV static. It's highly complex, but totally meaningless. But specified complexity, that's where you have high complexity that conforms to an independent functional pattern. Digital software, as a perfect example. Unguided nature cannot produce that. It strictly demands an intelligent mind.
So, when we look at DNA, we aren't just looking at a mere chemical pattern. We are literally looking at specified complexity. DNA is a digital, programmatic code.
The chemical bonds do not dictate the sequence of the bases any more than the magnetic material of a hard drive dictates the software written on it. The sequence is entirely arbitrary.
To put it in perspective, the information in a single human DNA molecule would fill a 36-volume encyclopedia. Code is imposed on matter. It is not generated by it. And philosophically and mathematically, a code demands a programmer.
Furthermore, even if you somehow have the code and the proteins, they have to be organized into functional biological machines. This brings us to molecular biologist Michael Behe's famous concept of irreducible complexity.
This refers to a biological system made of well-matched, interacting parts, where the removal of any one part causes the whole system to cease functioning. These microscopic molecular machines violently defy step-by-step evolutionary construction.
The classic analogy for irreducible complexity is your standard mechanical mousetrap. It's got a spring, a hammer, a catch, and a platform.
Now, if you remove the spring, you don't just catch fewer mice, you catch 0 mice. You just have a totally useless pile of parts. The biological reality of this concept is found in microscopic structures, like the bacterial flagellum.
The bacterial flagellum is literally a microscopic rotary biological outboard motor. It's constructed out of about 40 distinct proteins, forming a rotor, a stator, a driveshaft, a U-joint, and a propeller.
It's mind-blowing. If even one of those proteins is missing or deformed, the motor offers absolutely 0 survival advantage. It doesn't work a little bit. It is entirely broken. Natural selection would have discarded the intermediate, non-functional stages long before the motor was ever completed.
All right, part 3, mathematical probabilities in the academy.
We need to transition now to the staggering mathematics of random mutation, which Christian philosophers like William Lane Craig have highlighted heavily. We've got to observe the hard, unforgiving limits of unguided chance. Just consider the probability of forming a single average-sized functional protein of 150 amino acids by sheer chance. First, they have to link via specific peptide bonds.
And the odds of that happening are roughly 1 in 10 to the 45th power.
Second, every single one must be left-handed.
That is another independent probability of 1 in 10 to the 45th.
And finally, they have to be arranged in the exact specific functional sequence.
When you multiply those independent probabilities together, the odds of a single functional protein forming by sheer chance in a primordial soup is approximately 1 in 10 to the 261st power. Seriously, just let the sheer, devastating magnitude of that number sink in for a second. It is practically indistinguishable from absolute impossibility. To put that staggering number into perspective, consider this fact. There are only about 10 to the 80th power subatomic particles in the entire observable universe.
Even if every single particle in the universe was an amino acid, shaking and reacting at maximum speed, it wouldn't even come close to scratching the surface of the probabilistic resources needed.
But what about time? People will often push back and say, well, given enough time, anything can happen. Okay, well, there have only been about 10 to the 17th power seconds elapsed since the Big Bang.
The time elapsed since the beginning of our universe is microscopically small compared to the time required to generate life by random chance. The mathematical reality completely proves that the time solves everything argument is an absolute myth.
Which brings us to part 4, the pulpit, equipping your church.
Having done the heavy lifting in the academy, it's time for us to wrap up this explainer by moving right into the pulpit. How do we actually translate this heavy, intellectually rigorous science into everyday, life-giving faith for our congregations?
Host 1. Imagine a student in your youth group comes to you. They're fearful because their biology textbook implies they are just a cosmic accident. Your pastoral strategy here is to de-escalate that fear by educating them on the high-tech reality of the cell. Secular narratives portray the cell as just a simple blob of protoplasm.
Host 2. But you can show them that a single cell is vastly more complex than a modern automated car assembly factory, complete with transport trucks, power plants, and digital code readers.
This completely flips the script, turning intimidating biology into an awe-inspiring revelation of design.
And that allows you to directly bridge these scientific realities to deep, reverent worship. When David wrote Psalm 139, he obviously didn't know about DNA, ribosomes, or molecular motors, but he knew the Creator.
By teaching your people that our bodies don't just contain blind chemical reactions, but written programmatic instructions, you are reminding them of a beautiful truth. A program fundamentally requires a purposeful programmer.
So here is the homiletical big idea for your next sermon on this topic. You can look your congregation in the eyes and confidently say, every cell in your body contains a literary masterpiece written in a 4-letter digital alphabet. Your biology does not point to a cosmic accident. It contains the signature of an author. It is such a beautiful, scientifically grounded summary that directly touches the heart. We've journeyed from the skeptical myths of the street, through the rigorous halls of the academy, and finally to the sacred responsibility of the pulpit. The evidence of design is overwhelming, and it's undeniable. So I want to leave you with this final, provocative thought as a church leader. If every cell is a literary masterpiece, how will you introduce your church to the author this Sunday? Thank you so much for joining us for this explainer, and we pray you preach these truths with incredible confidence, clarity, and passion.